So thank you very much for coming. Welcome to the seminar. It's my great pleasure to introduce Dr. Sourjik from the Max Planck for Terrestrial Microbiology in Marburg, where he heads the director of the Department for Systematic Microbiology. And so his research area is the design principles of signal transduction pathway and gene regulatory pathways in microbes, like E. coli, yeast, I think, most of these model organisms. And so I know him from roughly from 2005, when he collaborated with my postdoc supervisor at Imperial at Princeton University. They worked on bacterial chemotaxis, and that was also the project I was working on. At that time, Victor was at the ZMBH at the University of Heidelberg. And before that, he was a postdoc in Harvard with Howard Berg, where he developed some really nice thread probes to look at signaling in living cells. So really cool stuff. Very excited that you're here. And over to you. That's the title. Very excited.
Yeah. Well, thanks a lot, Robert, for giving me a chance to speak here. I think it was already maybe 10 years ago that I gave a seminar here last time, but I was a year and a half when Robert was giving his inaugural lecture here to introduce Robert. But I'm glad that I also have a chance to talk about science now. So in our group, we are quite diverse in the range of topics we are interested in, but most of them are related to physiology, physics, and also, since more recently, evolution of microorganisms. And the topic I'm going to focus on today, I think, nicely combines all of this. And this is a very, very, very general question, whether we can understand their cellular networks or generally biological processes as products of evolutionary optimization. And for that, you might actually know this famous phrase from Theodosius Dobzhansky that nothing in biology makes sense except in the light of evolution. I think that's very true. And by the way, when researching for this phrase, I realized that he was born in the town next to the village where my father's family in the Ukraine is coming from. So it's small world. So if you want to understand or even to address this question of evolutionary optimization of a network or a process, what we need to know is not only their molecular composition and the basics of a network, we also need to understand for what was this network actually optimized. So we need to know physiology of the pathway, say if you talk about the signaling pathway, not only in the lab, but ideally also in the natural environment where this network has been shaped by evolution. We need to know historical constraints of the networks, because essentially what evolution does, and maybe it's not that different from what also us humans do, is primarily tinker with the pathway. So nature keeps improving or tuning the process that has been already invented once, and maybe that's not that different again from, say, combustion engine, which been optimized for last 200 years. Right now, maybe will become obsolete at some point soon. But so that's the same thing with the natural optimization, right? If their particular signaling network has been invented once, it can be tuned to match one or the other task. Then another important constraint on the evolution of biological system, and I think it's particularly prominent in microbes, are the cost-benefit trade-offs. So what you typically find, if you look carefully, that biology cannot optimize the functions independently. So what is frequently there is essentially conflicts in optimization. So the nature can optimize or say enhance performance of one network at the cost of another network or process in the cell. So it's in a way kind of maybe analogous to, say, mass or energy conservation in physical systems. So there is kind of conservation of resources, or resource utilization in biological systems. And last but not least, what we need to understand, and I think that's not that much, at least recently, has not been that much appreciated in biology, that we also need to think about the physical constraints on biological systems. And I'll give a few examples later on that the performance of the system is really limited by physics. And again, this is particularly true in microbes. So I think this phrase from Theodosia Dobrzansky at least needs to be extended, that we need to understand both evolution but also physics of the system. And microbes are really nice model systems for that. And as Robert mentioned, we primarily work with E. coli and budding yeast, like two best understood and most studied organisms. Just we saw their genome of E. coli outside of this lecture room. And the advantages of microorganisms are several-fold. First of all, they're relatively simple. I mean, they're still amazingly complex, but certainly more simple than higher eukaryotes. They're, at least sometimes, the functions and constraints on their networks are better defined than on the eukaryotic networks. And maybe most importantly, microbes had much longer evolutionary time span of optimization. So for non-microbiologists among you, for most of its history, our Earth has been populated exclusively by microbes. So, and they also have typically shorter generation times, that higher organisms, meaning that evolution went through thousands or millions of cycles of optimization of a particular network in microbes, which is certainly not true for higher organisms. And so our most favorite model in the lab is motility and chemotaxis of bacteria, and specifically E. coli. And I guess you know the basics of bacterial motility from Robert's work, and also from maybe the biologist among you from work of Morgan Beebe in here at the Imperial. But just a quick update. So E. coli, like most other bacteria, can swim by rotating helical filaments. And E. coli has several of them, and you see them nicely from a movie that's been produced in Howard Berg's group, and I was a postdoc there. So they're here filaments, and the cell bodies are labeled with a fluorescent dye, which is non-specific, essentially labels all the amino groups at the cell surface. And then you see that kind of swimming of bacteria here can be roughly separated in two phases. Sometimes this flagella form a bundle behind the cell, and this propels the cell forward. And in other instances, this bundle falls apart and the cell tumbles. And these are associated with the two directions of rotation of flagellum motor, and which is itself already a very formidable molecular machine. I would say requires 40 different proteins to be constructed, spans their entire cell envelope, and it's been powered by proton flux. So it's like an engine. Protons flow in, and the stator complexes rotate with which proton passes through, and this induces rotation of the rotary part of the flagellum, and this is then transmitted to rotation of this filament, which is substantially longer than the cell itself. And here is a cryo-EM structural reconstruction of flagellum motor. Again, you see that this is a very, very beautiful machine. And it also includes, maybe I should mention it here, an export apparatus. So it's also evolutionarily related to so-called type 3 secretion system. So this is the secretion system bacteria use to translocate effectors into mammalian cells, for example. So it's also related to infection systems. And this is their export machinery as the base of the flagellum. And all the subunits to construct this lone flagella has to be transported through the core of the filament. So essentially, flagellum is assembled at the outside, so at the distal tip. Now we understand pretty well not only molecular biology of flagellum biogenesis, we also understand the physics of swimming, and again, this is large extent due to the work of Howard Berg, who was amazing postdoctoral mentor, regretfully passed away about a year and a half ago, but in the very respectful age of 88. So we know that their cell body propulsion is essentially induced by thrust generated by rotating flagellar filaments, because of the helicity of the filament, it pushes at a liquid at an angle, and this essentially creates a force along the long axis of the flagellar bundle, and this can generate a thrust to push the cell forward, and it can be mathematically described by so-called resistive force theory. We also know the basic pattern of motility for E. coli or for other bacteria where it looks fairly similar. So this alternating periods of running and tumble generate something that looks very similar to a random walk. So this is the simulation we did long time ago of bacteria with actually pretty realistically moving, released at one point here, and then they're spreading in the environment, which again looks like normal diffusion, can also be described using the same equations as used to describe diffusion. And so this kind of diffusion-like behavior can serve several things or tasks. It enables bacteria to generally explore the environment. This is relatively slow, as we know that diffusion is not very efficient over long distances, but local exploration works pretty well. It is also important for interaction with the surfaces. So actually here you see several cells are stuck to the surface. That's probably not really specific interaction, but we know from other experiments in other groups and in our group that swimming can largely enhance bacteria interaction with the surfaces, because it enables them to overcome the repulsive force, so they can attach to the surface much better. There are also other interesting behaviors that motile bacteria can exhibit, for example, collective motion. So swimming bacteria can also hydrodynamically interact with each other and form this kind of beautiful rafts or packs of cells moving around at higher densities or the surface, for example. And probably most important part of the function of motility is to enable chemotaxis. And chemotaxis is again a behavior which is very well understood and became one of the best and most thoroughly analyzed models of simple behavior. So it describes the ability of bacteria or other organisms to follow chemical gradients in the environment. And the way bacteria is doing that is by temporal comparisons, and that's again Howard's contribution to the field to describe this strategy of so-called biased random walk. So bacteria choose a swimming direction randomly, or more or less randomly, and then compare environment and time, because they're simply too small to be really able to measure gradient along the cell body efficiently in space. So they rather kind of extend the comparison length by measuring their environment and time. And then if environment is getting more favorable, then bacteria suppress tumble, so they keep running in this direction, or even if they tumble, they only tumble a little bit, as shown here, to kind of try to find even more favorable direction along the gradient. And this strategy of temporal comparison is fairly efficient. Again, this is a simulation, but realistic one, of same bacterial population spreading in a gradient. So gradient is pointed towards the right side of this simulation field, and you can appreciate that spreading in this case is much faster than in the previous simulation I And in addition to this tumble control, what bacteria also need is a short-term memory. So essentially, they need to be able to remember their environment because they compare in time several seconds ago, and this is done by molecular system that methylates receptors. The receptor covalent modification of receptor serves as a memory to remember the past condition for several seconds. And I mean, generally, there molecular pathways that mediates chemotactic behavior is also very well understood, and no new proteins, at least in the coli, has been found in the last 40 years, I think. So essentially, we know all the functions pretty well, and how signals are transduced here, and again, you might know from Robert's work, who also contributed to that. The receptors associated with the kinase, which then phosphorylates TY, which is a small protein, can diffuse and then phosphorylate it and bind to flagellomotor, and this induces their switch of rotation in flagellomotor from counterclockwise to clockwise, and this induces a tumble. So QI, phosphorylated QI is a tumble generator. And then in addition, you have a phosphatase KZ, which is a counter pair of the kinase, and you have another pair of the enzymes which oppose each other, and these are, again, this receptor methylation system, which serves as a temporal memory and also allows bacteria to adapt to the particular environments. And actually, these pairs, as a side note, of opposing enzymes are quite important. So we worked on this later, a while ago, to show that these pairs can serve the function, can enhance robustness of the pathway, because essentially they counteract each other, and this enables the cell to compensate the effect of gene expression, noise, and temperature on their output of the pathway. And yeah, so and chemotaxis, as I mentioned already, can enable bacteria to follow gradients and spread in the environment, find the sources of nutrients, for example, or avoid harmful conditions such as repellents. So we know molecular biology, we know their physics, we know the basics of pathway behavior. Another thing that I mentioned already before, which is important to know to better understand evolutionary kind of optimization of the pathway, is the physiology, so what's the pathway actually doing in the natural environment. And for chemotaxis, even in the coli, it's surprisingly not that well understood, but there are probably three areas where chemotaxis plays an important role. One of them is nutrient acquisition. So again, many of their chemoeffectors are nutrients. Another one are collective behavior. So chemotaxis also plays an important role in, say, formation of biofilms and other collective behaviors of bacteria. So bacteria can also secrete chemoeffectors that attract other bacteria in the group and enables them to form aggregates, kind of also nice example of self-organization. And last but not least, their chemotaxis and motility are also important for host microbe interactions. So bacteria can use gradients emitted, for example, by the plant roots or by epithelial layer in the intestine or by some like particles of marine snow or eukaryotic organisms in the ocean to be attracted to them and to undergo either beneficial or pathogenic interactions with the host. And this different types of behaviors where chemotaxis plays a role might also put different optimization constraints on the system. Right? So then optimization for collective interaction is not necessarily the same as optimization, say, for finding nutrients. And so in the rest of my talk, I'll focus on this particular physiological function of chemotaxis, which is probably also the most important in bacteria at least. And again, as I mentioned, an evidence for that major attractants for E. coli are amino acids and sugars, and you even see clear correlation between nutritional and chemotactic preferences. So, say amino acids, which are most valuable metabolically, nutritionally, are also the best chemoeffectors, and the same is true for sugars. And the function of chemotaxis and motility in increased nutrient uptake is obvious, right? If they find bacteria can use chemotaxis and motility to find higher concentration of nutrients, they could benefit for their growth. Now, on the other hand, so we need to consider the costs of motile behavior, and they are substantial. Again, I mentioned that this is their flagellomotor is a huge structure, and there are several of them in the coli. And if you count the amount of proteins that goes into biosynthesis of flagella and also in the chemotaxis system, it's several percent of the total protein budget of the cell, which is quite substantial. And a similar calculation can be done for amount of energy spent for motility, and that is again several percent of their proton gradient. This is dissipated by a bacterial cell, goes into swimming. That's why their expression of motility genes is tightly regulated, also organized in several classes, and separated by special checkpoint. So essentially what happens is the base of flagellomotor is synthesized first, and this is already large investment, but not the major investment, because most of the protein will go into the filament itself and in the chemotaxis system, and these genes for flagellar filament and chemotaxis system are only expressed when the base of the motor has been already synthesized, and then this negative controller anti-sigma factor, FGM, is getting secreted through the pore. So the pore normally secretes flagella subunits, but it also secretes this negative regulator, and only when the export apparatus and the base is constructed, negative regulator is removed from the cell, and positive regulator of this class 3 genes is active, becomes active, and the synthesis takes place. So it's quite elaborate system. So given that potential benefits and the costs and complexity of regulation of the system, we need also to think about the trade-offs. Right? So essentially, under, say, particularly poor nutritional environment, bacteria can use chemotaxis and motility to find better nutrients, but they also pay a price. And then the question is, what is the balance between the price and the benefit? And kind of nice illustration of that comes from experimental evolution of motility system, which can be done relatively simple. And this was the work by Bini, a postdoc in the group. So you can take wild type strain, particular laboratory wild type. We have been working with that back then, and you can let it spread on this soft agar plate where bacteria can swim through the pores of the environment in the agar and follow chemotactic gradients that they create themselves. And then you can keep re-inoculating, taking back bacteria from the edge, inoculate again, and go on and on, kind of classical design of experimental evolution. And then what you see indeed after 30, actually already after 15 passages of cell, bacteria substantially improve in their spreading. And this enhancement can be about 50% or so. And you can also synthesize. Yeah.
What's the unit of the y-axis?
Spreading compared to the wild type. So I guess one, it just normalized to the wild. Normalized to the wild type, to the original strain, essentially. This divided by this.
Is measured as the radius of the spherical?
Yes. Which can also be So essentially, spreading is linear with time. So you can also you'll get more or less the same number if you measure the rate of spreading. But yeah, it's a legitimate question. So we looked at that. So it's a spread. So it's more or less the same.
So where's the source of nutrients in that experiment?
Yeah. So the source of nutrients they create themselves. So essentially, what happens when bacteria colony starts growing? So it's a rich media in this case, where TB tryptophan, broad soft targets, so a mixture of amino acids. And when bacteria start growing, they degrade nutrients in the middle, and then this creates a gradient to the outwards. And since they can swim in the soft targets, they chase this gradient. So if you do the same for non-chemotactic strain, they can spread a little bit, but they spread much less efficiently, and then it's not really linear as in the case of the wild type, where essentially it's a wave, like traveling wave, that you get spreading outwards. But it's a combination indeed of metabolism, motility, and chemotaxis. So it's all and growth. So essentially, it's all important. Then we can also look at the mutations that's been published already some time ago. So just briefly, that we get here. Actually, to our surprise, what we saw that most mutations we got were in the export apparatus and their flagellar base. But that's why I mentioned the anti-sigma factor. So what we think was happening, or is happening actually, in this mutant, that they increase the efficiency of their negative factor secretion from the cell. And at the same time, this also increases the efficiency of flagellin secretion, so they can assemble flagella better or longer flagella. And they can also secrete this anti-sigma factor, and then this releases the sigma factor, and this upregulates flagellar motility. We're kind of wondering why it happens this way and not, say, for example, by introducing mutation in anti-sigma factor or increasing expression of sigma factor. But then I remember there were quite inspirational talks by John Doyle. I don't know whether maybe familiar to some of you, he's an engineering professor at Caltech, very bright guy. And he was proposing that many biological networks have a structure of a bow tie, where you essentially have like complex processes upstream, downstream, and then you have there some like intermediate process which connects the upper and the lower part of a bow tie. So I think something like that is also happening here. So you have there essentially flagellar export apparatus that consists of many proteins, and essentially you can mutation on the modified 5-6 proteins to increase the efficiency of anti-sigma factor secretion, and then downstream again it regulates a large pool of genes. So it might be, and maybe actually even shown by simulation, that this kind of bow type structure has higher evolvability. So it might have been selected for nature to be able to tune it more efficiently. And so another nice result of this work was this beautiful trade-off line between growth and swimming. So it's a swimming velocity, or you can also plot gene expression, but then it's not a nice line. then it's more like hyperbolic. So here, if you plot swimming velocity as a function of the growth fitness, or growth fitness as a function of swimming velocity, then and here's the wild type, then what you see, all of the mutants we were getting, and even individual mutations, so individual single mutations, when we cross them in back, they move along the same line, with a couple of exceptions. And also the knockouts, for example, of flagellar genes which do not synthesize any flagella at all, or fly-C knockout, the flagellin knockout is also more or less on the same line. So it seems to be a really very nice trade-off line between growth and swimming. That's which I think illustrates the point of the growth motility trade-off, fitness trade-off.
Just one question.
Yeah.
I guess then here the nutrients are not really limiting, right? Because
Yeah.
they would need
Yes.
chemotaxis to find them in the first place. It would maybe look at different different.
Yes, yes. So this is really without any benefit. So that's in the liquid with a shaken. So that's only cost as a function of motility. Yeah.
Is there a theory why the trade-off should be linear?
Good question. I don't think so. Why? So again, so if you plot it against gene expression, it's not linear. It's more like hyperbole, right?
I mean, it comes out of the Terry-Vars theory, but it's a straight.
But why is it linear with swimming? Yeah, so in principle, where trade-offs come from, right? This is maybe just quickly to mention here that there were also other two papers around the same time doing similar experiment in different backgrounds under different conditions, and they saw a similar trade-off. They actually got different mutations, which was interesting, but essentially phenotypic changes were same. Genotypic changes were somewhat different because they used different backgrounds and different conditions. Yeah, so this is what Robert was mentioned, where we also collaborated on their motility growth trade-off, and this is it was inspired by Terriquois' idea of proteome partition, which I guess some of you are familiar with. I assume so. Their idea is simple. It's a kind of also conservation law, you can say, in bacteria, that the number of proteins you can actually put in a bacterial cell is limited, and bacteria is indeed extremely full. I think about a third of their space in the bacterial cell are proteins, so there is not much more space. And you also need some space for the chromosome and for RNAs, so essentially there is no free space, more or less, in bacteria. So meaning that if you want, you can divide proteome in several sectors, and if you want to increase the size of one sector, you need to decrease the size of another sector. There is no way around it unless you change the size of a bacterial cell, which has its own limitations as well. And the idea here why motility can affect growth is essentially if you increase the size of the sector for chemotaxis motility, then you would decrease the size of the sector which is responsible for protein biosynthesis, ribosomes. Right? So that's essentially, in a nutshell, the nature of this trade-off. Now, that doesn't really answer the question, why is it linear with the swimming velocity and not with whatever gene expression, right? Yeah, we can talk about this later. So in this direction, there is a trade-off in this direction. One can also see it in an opposite direction, that how their number of fraction of motility and chemotaxis genes in the proteome would be affected by different growth conditions, right? And this is related to classic catabolite repression, which some of you, I guess, biologists among you, but I guess also systems biologists among you would probably know that as classical examples from Monod in the 60s already. What he observed back then, that some genes, say for transporters, for lac transporter, for example, are repressed by glucose, by good carbon source, right? And this can be generalized, as has been also done by Terriquois, and essentially saying if the cell has a good carbon source, wants to grow faster. To be able to grow faster, it needs to increase the fraction of ribosomal genes, right, to synthesize proteins, and also ribosomal RNA. And this has to push against the other sectors of the proteome, and then you have a constant sector of the proteome which is needed for homeostatic functions in the cell. And so the fraction you can decrease is the metabolic fraction, so for some non-necessary metabolic pathways, which maybe are not required if you grow on glucose. And what we kind of assume that motility and chemotaxis would also belong to this fraction, because in principle it has a very similar function, right? So here you have alternative pathways for some nutrients, metabolite utilization that you can vary dependent on the growth conditions. And motility and chemotaxis do kind of the same. So they also increase availability of additional nutrients. And indeed, if we assume that, and this was worked by Robert, then we can get nice dependence of expression of motility and chemotaxis genes on the growth rate. So here's the growth rate compared to, well, it's just actually per hour. And this is a model compared to experimental data. So that's actually, the model was not fit to the data. That's really prediction from this proteome partitioning model. So indeed, if E. coli grows in a poor carbon source, say ribose, it expresses much higher levels of motility genes than if it grows in glucose in a good carbon source, as again expected from this proteome partitioning model.
So what does the slope depend on?
So, slope.
The slope of this line?
Robert. You need the model.
Sort of one of these lockdown papers, nothing better to do this. And anyway, there's these sort of uptake coefficients and so on, which are from Terry-Bars, essentially how quickly nutrients are taken up and also converted to nutrients, I think. So there were some sort of phenomenological parameters, and they sort of work very well for that scenario. I don't know if that answers your question.
Yeah, essentially there is certain mass biogenesis, right, per unit of time, which is related to the growth rate, and then I guess you need that and that many. So you have efficiency of protein synthesis by the ribosomes, which then kind of autocatalytic, right? They also synthesize themselves, and then they synthesize the rest of their of the proteome. So if you know total amount of protein you have in the cell, and you know the efficiency of the ribosomal fraction, then I guess you get, oops, sorry. Yeah. Okay, so going beyond the gene expression, so what else depends on the growth rate? In a similar way, we see increase in flagella number at low growth rate, increase in the also flagellar length, and also in swimming velocity. So essentially, it's not only that flagella expression is elevated at poor nutrients, or say in poor carbon sources, but indeed motility is enhanced proportionally. So that's all as expected. Then if we think about cost-benefit trade-off, we want to be able to measure both, and we can in E. coli. Again, E. coli is a nice system for that, and we can do it by using flow cytometry, where we just core culture, say wild type with different knockout strains. So FLHC synthesizer expresses no flagella genes. So essentially this gives us the value of the cost in absence of any chemotaxis benefit. Or we can do experiment in presence of chemotactic gradients, where E. coli also indeed can benefit from chemotaxis, but then we compare the wild type with KY knockout. KY was the stumbling generator I mentioned. So it still swims perfectly well, but it doesn't do chemotaxis. And then we can, by using two colors, we can then separate the cells in a flow cytometer and compare their numbers of wild type against the knockout and the different growth conditions, right? And what you see here, for example, in glucose, their numbers are comparable, say, but in the ribose, where flagella expression is higher, the wild type is almost completely outcompeted by the knockout strain, which synthesizes no flagella, right? And thus we can plot as a function, again, of the growth rate, and indeed, in poor carbon sources, the cost increases a lot. And this is really the function of expression level. It's not that in ribose somehow flagella are more expensive per se. Is because flagella are expressed at higher levels that are more expensive. Because if we have the strain with a constitutive high expression of flagella, then the cost is also high even in glucose. So it's really a question of expression and not of the carbon source itself. Now, we can also look at the benefit. Again, so in this case, we compete wild type and against non-chemotactic but motile strain, which pays similar cost, but gets no benefit. And then we provide bacteria with a source of amino acids in the medium to which wild type can accumulate and profit from high nutrient abundance. And what we then see is also nice, that in the poor carbon source ribose, there is indeed high benefit of being able to find additional nutrients, amino acids in this case, and there is essentially almost no benefit finding additional amino acids if the cells are growing in glucose, which is already an excellent carbon source. And in this case, actually, it's not that much the expression level, because even the constitutively high expressing strain doesn't get much benefit. It's only slightly higher than the wild type where expression is low. And here is essentially the open symbols and closed symbols follow each other, which means to us that it's really the question, in this case, the question of nutrient source. If you have poor nutrient carbon source, it's important to find additional nutrients. If you have rich carbon source, good carbon source, then it's not important. And what is nice when we plot their expression level against their chemotactic benefit, we get a straight line essentially, which to us indicates that E. coli indeed invests in expression of flagella genes in proportion to their anticipated benefit. It can obtain if the gradient is available. So it's not causal relation, because even under conditions where in a shaken flask in a culture, where it has no benefit whatsoever from chemotaxis gradient, it still invests. So the expression level doesn't depend on the presence of the gradient, but it is proportional to the benefit that E. coli would obtain if the gradient of additional nutrients were available in the medium. So it's really an anticipatory investment, which again, proportional to potential benefit. So next question we ask, again, this was also published a few years ago. So next question we ask, and what we are investigating now, is about absolute levels, right? So in the slide here, maybe I go back. So in the slide here, essentially we see proportionality, but that tells us nothing about the absolute level of expression. So how much expression in some absolute units is actually optimal. And so to address this question, the project has been taken over by Ira, PhD student, has placed flagella regulon, so the entire regulon, under inducible control. So we can tune by levels of IPG. And so this allowed us to actually measure both benefit, or at least performance in this case, of swimming, and the cost as a function of flagella expression levels. And what we saw is actually quite nice, I think. So with increase in expression of flagella genes, motility initially increases, but then at some point it saturates and high levels. And if you look where the wild type is, it's already kind of indicative, right? Why the wild types could be there. And indeed, this is not only, at least among laboratory strains we are starting from, special form, this MG1655, oops, sorry, maybe I should, which is our standard or more standard E. coli strain, but we can also look at other E. coli strains, and they have similar levels of flagella gene expression and similar levels of motility.
Is this, by any chance, related to the question I was asking, that because the theory said that gene expression and growth rate, the gene expression of the flagella sector is kind of linear, and here you're showing velocity and gene expression are linear, so
Well, just
I guess it's suggesting you're not at the plateau part of it, but maybe at the medium part of it.
well, I'll get to that, so maybe we can wait with the discussion until a few slides down the road. So question is, I guess what Wahid is also into why does it saturate, right? So, right, why? But here it's not related to growth, right? So here growth doesn't play a role. I mean, there is obviously at high expression, so I'll show it also later. There is some effect of growth, but it's not. It's relatively minor. But the question is why E. coli doesn't swim faster, even at high expression of flagella genes. And one trivial explanation might have been that E. coli cannot do much with more. Maybe there is a limit of flagella proteins that it can make use of, and if it has too much of that, maybe it simply cannot build more flagella for whatever reason. That doesn't seem to be the case. So we can also, again, nicely stain flagella filaments and count them. And what we see indeed, number of flagella filaments keep growing at high expression levels. So this is a wild type, which is around 5, as it should. But if you increase expression of flagella genes even higher, we also get more filaments. Meaning that cells here actually have indeed more flagella, but they don't swim faster. Also, flagella seem to be have the same length. So if you look at the number, dependence of number of flagella on the number of amount of the external flagellin into the media, or I say outside of the cell, so essentially all the flagellin protein here, it's pretty linear, suggesting to us that the flagella are essentially the same. So they have the same length, and just the number of flagella increases proportional to the amount of flagellin.
So is the saturation due to a limitation of the experimental media? Because the media is the soft agar, right?
No. This is in the liquid. So that was in the liquid.
Oh, it's in liquid. So
That's in liquid.
Okay.
Which I guess I also, well, so the next slide. Essentially here, right? So that's if you then plot the flagella swimming velocity or bacterial swimming velocity as a function of flagella number in the liquid. So then it kind of shows a saturation function. Question is why? I mean, here resistive force theory that was exercised by Remy Collin, an excellent biophysicist in our department. So if you put down the numbers in resistive force theory, you do get a saturating function. The absolute levels are here somewhat higher than what we measure experimentally, but this is a very relatively simple treatment of bacterial swimming with multiple flagella. But essentially you get a saturation with the number of flagellar filaments, and reason for that is essentially that if you add more flagella, they increase the thrust indeed, but at the same time you also increase the drag force on flagella filaments themselves because you have multiple flagella, and this takes away much of the additional thrust flagella generate, and that's why you essentially get a saturating function with the number of the filaments.
I mean, is there also some interference, right? If you have too many flagella, which is not part of the theory, I suppose. You know, it might even flatten off earlier. You mentioned, the level doesn't fully match.
Yeah. So this is a very simplified treatment, which assumes that all flagella are independent. So actually, doesn't even consider bundling. So it assumes all flagella are independent, and that each flagella contributes essentially thrust, but also contributes the drag as increasing swimming velocity. The more precise treatment would require really to consider effects of bundling, effects of energy dissipation from flagella, essentially interacting with each other in the bundle, and rotating probably also interactions of flagella with the cell body, also dissipating energy. So I just say, brainstorming in this very simple model, you could actually get some kind of saturating function with the number of flagella. So it looks like this saturation we see experimentally is really due to the physics. So then bacteria, or at least E. coli, cannot benefit much from having substantially more flagella than the wild type cells have already. So it looks like the number of flagella E. coli has is relatively optimal. Now we get back to the cost question. So if we measure the cost under these conditions of flagella expression, right, so we get, again, relatively straight line, and the wild type is here somewhere in the middle. Meaning that if we increase expression, again, we keep increasing the cost, but we don't really improve the function any further because it's saturated already due to physics, right? And then if you kind of plot both these functions together normalized to the wild type in both cases, you get nice crossover, which is really indicating where the wild type should be if we assume that the wild type has been optimized to maximize motility while minimizing the cost, right? So that's exactly the point here. And this actually reminded me of the paper I was reading years ago about evolutionary optimization theory, which has been in the 80s and in the 90s, I think pretty hotly debated in evolutionary biology field, how much can we understand biological systems as products of evolutionary optimization. As a proponent of that was John Maynard Smith, who was, I guess, in UK in Sussex, was an engineer and biologist, and he was really believer in evolutionary optimization, but there were also papers by other evolutionary biologists who were saying this is all wishful thinking, and we cannot really build convincing examples of evolutionary optimization. But so example was John Maynard Smith was bringing was completely unrelated from that, was about birds chasing insects, lapwings, and the curves of the benefit and the cost of how far a lapwing would need to fly to catch an insect, and how much would it increase the benefits of flying farther would increase the benefit but would not increase the benefit linearly, whereas the cost would increase linearly, and then in the end you get such kind of crossover, and then the optimal how far bird would need to fly would be somewhere here. Depends on the relative importance of catching an insect, and the cost could be point where the difference between benefit and the cost is maximized, but it can be essentially anywhere here, dependent on the limitations, right? And that looks, in a way, superficially similar to what we see for the cost-benefit trade-off in bacterial motility. Now we can also look in the minimal media, so related to what I was showing before, and this here is a bit different, so it's not the same as in the rich media. Before, so first of all, for whatever reason we don't understand well, their swimming dependence on expression level is in the minimal media is below the one we get in the rich media. So for whatever reason, bacteria with the same expression level actually do not swim quite as fast if they are grown in the minimal media. Also, that's the dependence essentially is the same for different carbon sources. So there's some difference. And another difference we see the cost is also higher in the minimal media. Again, regardless of the carbon source, the cost of expressing motility genes at the same expression level is higher. So in this case, you don't really see this nice crossover. What you get instead is essentially both curves kind of going in parallel for cost and the benefit or function of motility. And what we also see that the wild type is, in this case, indeed does not maximize motility. So the wild type expression levels, as I showed before, depend on the carbon source, but in either case, they are somewhere in the middle of this curve. So for succinate or ribose, the poorer carbon sources are higher. For glucose, they're lower, but they are not maximizing. So what happens here? So what might be the reason for fixing particular expression level? And here again comes their experiment with measuring the benefit of chemotaxis. Actually, the scale here is different because these experiments have been done earlier and was a different plate reader that was used to read out flagellar expression, but it's the same reporter. And what we see again, this is the cost, right? That's similar dependence as I showed before. But then if you look at the benefit, what we see is a benefit as a function of expression in succinate, poor carbon source, saturates at a higher level than in the glucose, in a good carbon source, right? And this is native expression levels here shown with a dashed line. So what it suggests to us that, again, connected to anticipatory investment, I showed before, it's not only the relative level, which is tuned according to anticipated benefit of chemotaxis, but likely even the absolute expression level is actually tuned according to anticipated benefit. Okay, and then last few slides, we also looked at their natural isolates, right? This was all about the laboratory wild type, K12 strain, again, standard reference, wild type E. coli. What about E. coli isolates you get from patients or from nature environment? And there is a nice echo collection for those isolates, and we looked at most of them, and substantial fraction of them are motile. But if you look at their expression level and swimming in this natural isolates, they're all kind of here below mg, so that's mg, our reference strain, right? And they're all have expression levels and motility roughly on the same curve as we saw for mg strain, but lower than mg expression. And this again brings me back to this kind of general optimization curve that I showed before from John Weiner-Smith. Again, the optimal would be somewhere here, and this is exactly what the natural isolates are doing. So they are distributing here the expression level and motility in this area where nature should optimize motility dependent on particular environment. What we observe, so when we put the same strains on the soft agar plate against the Sagar assay we used for evolution before, then all of a sudden most of them became as good as mg. So this is a liquid, right? The swimming, and this is their strident, which is also motility dependent in the soft agar. So this all of these guys actually were going up to higher motility levels in the soft agar. It could be maybe even nicer illustrated here when we plot strident on the soft agar as a function of velocity, motility in the liquid. So the group of strains are similar, like mg, strident and swimming similar to each other, but there are a group of strains which only swim in the soft agar and actually do not swim at all in the liquid, and also another group of strains where motility is enhanced in the soft agar. And this is an interesting phenomenon. We can actually describe it a couple of years ago for several isolates that indeed, you can see an example, doesn't swim in the liquid. So this is mg, you see tracks. This is one of their pathogenic E. coli isolate. It doesn't swim, you put it on soft agar strain plate, it actually spreads. And you also see the gene expression is activated. So this is gene expression in mg, it's the same on soft agar and in the liquid. In a pathogenic E. coli isolate, motility is activated.
So why there are multiple ratings in your agar channel?
Classical chemotaxis, yes. So that's been already addressed in the 69, I think, by Yunus Sadler. Yes. So this is the outer ring is a chemotaxis to serine. So it's individual amino acids with their chasing. So there's a gradient of serine. The first amino acid E. coli eats up and then chases. And the second ring is aspartate. It's the second best. Sometimes you also see additional rings. These are other amino acids that are chased by E. coli, but yes. But this is kind of a really characteristic feature of chemotaxis. And yeah, so with this activation of motility in soft target, actually back then, because we primarily observed this for pathogenic isolates, our interpretation was that the E. coli activate this pathogenic E. coli can activate motility in the mucus, which might help it to infect the host. And might still be true, but it really seems to be very common among the natural isolates. So maybe this is also a resource saving strategy where E. coli, if it's somewhere in the liquid under conditions, maybe there's much turbulence and gradients are not stable. It's not beneficial to actually swim on the time, but at the moment it's in the intestine and the gradients are more stable and predictable, it activates motility to benefit from that. So that's at least could be a hypothesis. Okay, so with that, just summarizing, so I think could see at least three strategies E. coli and most likely other bacteria can use to optimize motility dependent on the environmental condition. So in the rich media where resources are not that limited, it looks like that E. coli, or at least our standard strain, maximizes motility up to the physical limit. But then later on, it reduces expression to what is necessary to minimize then the fitness growth effect. In the minimal medium, where benefit of motility is lower and the cost might be higher, the strategy seems to be different. Then E. coli really invests in motility proportional to what the benefit of motility might be, if the gradient would be there, of nutrients will be there. And then for natural isolates, we see that the distribution of native expression levels in this range, where again evolution can play with with optimal expression, probably dependent on the condition in which this or that isolate evolved in the nature. And some of them might evolve under conditions where motility is more beneficial, so the expression level is higher, maybe similar to our lab strain. Others might have evolved under conditions where motility is less beneficial and the expression level is lower. And for many of these isolates, again, I just mentioned, we see that motility is activated in porous media, which might be adaptation to the intestine, might also be related, that's something we are exploring now, to different dependence of motility on the number of flagella in a viscous medium. So that's resistive force theory could also predict under certain assumptions that having more flagella in a viscous medium would be more beneficial than. So the saturation point would be at higher number of flagella than in water at low viscosity. Okay, so is that finished? And yeah, so these are people whose work I talk about, and this is our group in 4D Marburg 2 years ago, and our collaborators on the project I mentioned, Robert, and also Hannes Link, who's at the University of Tübingen, who's doing metabolomics with us. And yeah, thank you for attention.
So there's a question, Ramesh.
I'm just kind of fascinated by the saturation view.
I think okay.
In the environment, the bacteria encounter phages, and flagella are landing sites for phages. I mean, since they're kind of evolved, and coming back to evolution theory, there's an optimum number of flagella that needs to be made so that the population is retained, so that the phage attachment has been minimized. Have you considered the possibility of that saturation that it exists because of?
Yeah, well, I mean, again, so it's a fully legitimate question. I mean, some of the natural isolates, right? Again, as I showed, right, have lower level, so do not maximize motility, so it's there somewhere in this area where they are motile, but not as motile as MG1655 as our reference strain. And we don't know why they have lower motility levels. but one of the selection they might have been exposed to is indeed their minimizing their risk of the phage attachment. I mean, it also might partly explain why many of them are not motile in the liquid. I guess the chances of encountering the phage is higher than somewhere in the mucus. So that's certainly one of the aspects of the selection pressure E. coli is exposed to. I mean, one can probably do it. I don't know, maybe that's also what your question relates to. One probably can also test it experimentally, right? So one can probably look at E. coli or evolve E. coli in presence of phages on, say, soft agar plates.
Well, if you have a mutation in one of the flagellar component where the phage lands, that's a comparator to the wild type. And see how.
Right. But then again, then I think the trade-off would really depend on the relative. I mean, I guess the theory would be easy to do it for that, right? You can just simulate what the optimal expression of motility, assuming there is some benefit, say, of finding a nutrient, and at the same time some risk of being lysed by a phage, right? And I guess depends, right? If the risk to be killed by a phage is very high, then the best strategy would be non-motile, right? Obviously, if the other way around, the best strategy would be motile, right? But then their actual expression level would depend on this relative risk.
Can you just explain again for the people who don't maybe know so much about it? I thought lambda phage is attaching to the LAMP-B receptor, but it's also
So there are also phages that attach to flagellar. So actually quite funny because then they use flagellar rotation to slide along the phage to find the cell body. Even so, that's a quite refined strategy. So yes, that's.
I suppose some of them also kind of use flagella as decoy. They kind of detach it, right? So they kind of can they release it into the medium?
Yeah, it depends on bacteria. So E. coli doesn't really do it, as far at least as I know. Some other it has been reported that some bacteria can actually detach flagella.
So decoy it for the phage, right? So.
Yeah, I don't know how efficient it is. I mean, it will be obviously would work on it very low titers of phages, right? Because I mean, how many flagella can bacteria release? So I don't think it has been. It will be very efficient decoy, I would say. So I think release of flagella might be for different reasons than bacteria simply, if they don't need it, they don't want to rotate them and don't have to dissipate energy. That's maybe more important than placing them as decoys for phages. But
it's quite late already, but any other questions? I mean, we certainly have room for a few more minutes. No more? Okay. So thank you very much. You answered already a lot of questions. Already a lot of questions during your talk as well. Yeah, thank you very much. Cool. Nice to see how it evolves afterwards, actually. Pretty cool.
my computer actually died, but because the battery died, but it's exactly at the right moment.
Because the first of the meeting scheduled.
Yeah.
So, okay.
Yeah, sorry, I went a bit long.